Good afternoon. I welcome all of you to the 30th lecture of our webinar series. We started somewhere in 2020, October. Now we have entered into the third year and we have been hosting a series of talks on various topics. And today we have Dr. Kalyan Shekhar Chakravarti from Ashoka University, who has worked on some interesting aspects and those kind of studies are very rare in India. I think he has made a very good study involving the ceramic samples. So first of all, I welcome Dr. Kalyan. Thank you very much for sparing your time to give a talk with us. And I request Umka to introduce the speaker and then we will continue. Yeah.
Welcome everyone for today's talk. So today's talk by Professor Chakravarti will be on multi-proxy reconstruction of human-animal interaction, a glimpse into the subsistence economy in Gujarat during the 3rd millennium BCE. Professor Kalyan Shekhar Chakravarti is an assistant professor at Ashoka University, Haryana, specializing in biomolecular archaeology and archaeological geochemistry. He is interested in reconstructing ancient animal herding practices and the utilization of animals and animal products to understand the role of domesticated animals and the interrelationship between human-animal environment in the development and dispersal of complex societies in South Asia, especially in India. So we thank you for your valuable time. You may now begin the talk, sir.
Okay, thank you, Professor Prabhakar, Dr. Sharda, and Umka for this nice introduction, and thanks a lot for inviting me for this talk. So yeah, I'll be primarily talking about human-animal interaction and how that shaped the subsistence economy in Gujarat during the 3rd millennium BC. So just to give you a summary of the presentation. So in this presentation, I'll be covering the history of human-animal interaction, a brief introduction to the 3rd millennium in Gujarat, the archaeological data and method that we currently use, the use of multi-proxy analysis using diverse archaeological data, our current understanding, and some unsolved questions that we need to pursue in the future or in ongoing archaeological endeavors. So the primary question that always comes to anyone working in archaeology or beyond is that why do we need to study human-animal interaction? Like, why is it that important? And it's pretty given that, you know, yeah, humans have been eating animals, using animals. That's very well and good. But what is the point of having this sort of interaction and an understanding of this sort of an interaction? So since the beginning, not only the current Homo sapiens species, but other species of hominin as well interacted with animals in a way that, you know, they have been interacting, and always through the history of human occupation, they have been interacting with its cultural environment. So that's a cultural and ideological structure at Göbekli Tepe. And then they were also interacting with their surrounding natural environments. So for example, land, water, animals, plants. And in order to doing so, they have been using multiple agents. And one of those agents are the animals, right? Now, when we talk about history of human-animal interaction, we have to understand whether we should consider animals as an agent through which humans were interacting with these various environments, whether it's cultural or physical or natural, or these animals were just raw materials that they were using for multiple purposes. Now, if we consider them as just as raw materials, then we can think of only their utilitarian purpose. But when we're talking about the interaction and animals as an agent, then it's not about only how humans were using them, but at the same time, how humans were procuring them, how humans were expressing animals and their interaction with animals, how humans were consuming these animals, as well as how humans were disposing these various animal remains. So since the beginning of human history, humans have been expressing their interaction with animals in multiple forms. Some of them are utilitarian, some of them are non-utilitarian, ideological, and symbolic. So on my left, I don't know if you can see the pointer. So on my left, that's this cave painting from an Upper Paleolithic site in Europe. So where you can see humans depicting animals. This is the animal engraving at Göbekli Tepe, which is in the Anatolia region. And here also we have been saying these are very early. So this is Upper Paleolithic. This is slightly pre-Neolithic, pre-Proto-Neolithic time period, where we have seen that animals were depicted in a very symbolic way. Then we definitely have this famous animal enclosure at Catalhayuk, where we can see bovine horns were placed, and there are arguments fairly recently that at Catalhayuk, even though they were cultivating and domesticating animals, there is clear signs of domesticated cattle and buffalo. But during the very early phase, they were most likely using cattle and buffalo for ritualistic and symbolic purposes other than utilitarian purposes, say, for example, as food. Okay, not only in other parts of the world, but we also have different sort of expression of animals via our ancient human. So this is an ash mound, Neolithic ash mound at Karnataka. So where we see a very different way of disposing animal remains. It's not a physical remains, but the animal dung that were burned and this sort of ash mound form. Then we have Harappans. These are the famous seals of Harappan. We know Harappan seals are full of different animal figurines. Somewhere the actual animal that you would see around, somewhere the ideological animal. So, for example, the famous unicorn. They also have pottery, and we have also seen depicting animals in the potteries. And then obviously the other major find of animal remains in any archaeological science are the archaeozoological remains, or the remains of animals that human consume. So as we can see here that humans have been interacting with animals and animal products in a very diverse way, which is not necessarily or entirely dependent on what they were eating. So it's quite clear looking into archaeological data from India and abroad that animal played a role or a major role beyond beyond as a primary source of food or traction or dairy or hide or wool. They also played a very crucial ideological and symbolic role. So when we try to understand this process of interaction, we have to understand it in its entirety. So starting from the selection and procurement of animal, right? So humans have been always making conscious as well as subconscious decision, which is either based on its climatic condition that the human is residing, the ideological conditions, their current understanding of the landscape, as well as their worldview, that they were choosing certain animals over other animals, right? Like any plants and any animals that you see, it is a clear understanding that there are out of all the plants or all the food that human can eat, human only eat 20% of that. So there are other eatable animals and plants around which we don't eat quite often. Say, for example, there are some animals and other animal products which we think are not suitable for our consumption. Say, for example, insects, right? So in some cultures, insects are considered to be not the ideal source of food, but in some cultures, insects are thought to be a very good source of food. And we also have similar evidences from India as well. And then once they choose the animal, then there is also a way to decide how would they procure the animal, right? So they would procure the animal live or they will procure the animal dead, right? So, and then there are different measures or different techniques or different methods of procuring those animals. There is obviously hunting, trapping, managing herd, domestication, and all sorts of things. Although managing herds and domestication are quite recent in human history, but even in the previous time, we have evidences of human hunting animals as well as trapping animals. And even we have seen that, the earliest evidence of domestication, which goes for dog, were primarily domesticated for purposes other than food, right? Although there are quite a few evidence that dogs were also consumed as food. Then once the animals were procured, there are also a very different set of interaction in terms of how animals were raised, right? So the seasonality of birth, if humans have specifically chosen a certain time when the birth would happen and the herd will give you birth, and then at the same time, they have also decided on what should be the male and female ratio in the herd, the nature of foddering, what these animals would eat if they were raising these animals, and we would see that, there are very clear decision-making processes, whether it is a utilitarian decision-making processes, which is a ideological or symbolic decision-making processes, but there are decision-making processes to determine what sort of fodder that these animals are going to eat. And then at the same time, access to water and pasture land, because this plays a huge role. When you have animals, the two things that you would need, the bare minimum, is the food and the water, right? So pasture land is a very important thing. And we have seen from the very early in the history, even to the very modern history, that how pastorals have been fighting for the pasture land, how they were being nomadic, how they were being semi-nomadic. We have seen how British rules were particularly catered toward pastoralism so that, the forest can be safe, so they can export all the nice wood that we have. Then at the same times, after they have secured the kind of animal they would procure, the kind of animal that they will raise and how they would raise, the next question comes to how they process the animals, right? So say, for example, when you have to determine the how would you process the animal would also be based on the kind of resources that you are extracting from those animals, right? So say, for example, are you particularly using that animal or procured that animal for meat, for milk, wool, hide, for its skeletal remain, for dung, or for companionship, or for any other symbolic meaning, right? So based on that, you would determine how would you process the animal. For some usage, you cannot have a dead animal, you need a live animal. But for some cases, you would need specifically dead animal and you cannot have live animal. Even though if you need dead animal, then there are certain things you will exploit from the animals, and there are certain things you would not exploit for the animals. And skeletal remain is a very interesting thing, because sometimes there are indications that there are suggestions that humans were probably exploring the dead carcasses in order to extract only skeletal remains. So they were not so interested about the meat, but they were more interested about the skeletal remains, because we have seen bones being used as a primary raw material for tools. We have seen skulls were placed in houses, for example, that we saw at Catalhayuk. So all these different sorts of resources that we can think of from the animal would also determine how the animal would get processed. And then finally, how the animal would get consumed, right? So there are multiple types of consumption. When we talk about consumption in any archaeological settlement, or even in the present settlement, or in the present culture, there are symbolic consumption of animal, right? Say, for example, the totems that we have. There are many animal totems that we use, and then we consume them symbolically. So not as a physical consumption, but a symbolic consumption. Then there are ideological consumption. Then there is definitely dietary consumption. So say, for example, when we are directly extracting dairy or meat from animals. And then there are also other utilitarian consumption, say for traction. Sometimes we need animals for certain type of activities, for movement, like horse-driven cart, cow-driven carts. Then we also have exploitation of animal consumption and animals for their wool, for their hide. And so different animals would have different sort of consumption tradition or consumptional traditions. And sometimes we have animals which they have like multiple different kinds of consumptions. And sometimes we have animals that were consumed for a very particular or specific purposes. Then the other thing that is also very interesting is that once this entire process is done, where does the animal ends up? Because that also would indicate that how were these animals consumed, or how was this animals perceived within this society, or the role that particular animal played in shaping human interaction with its cultural environment as well as its physical environment. So for example, if an animal of particular type of animals were only ending up as animal burials, if they were ending up in a garbage, if they were ending up inside the settlement, if they're ending up outside the settlement, if there are certain portions of animals that are only found inside the settlement versus certain portion of animals that were discarded exclusively outside of the settlement. So this entire process needs to be understood from whatever fragmentary archaeological data we have, in order to develop the as robust as possible type of human-animal interaction, right? our understanding of human-animal interaction across the globe is not complete, right? Primarily because we have a lot of fragmentary archaeological data. Our archaeological data is not complete. Very rarely we excavate a site in its entirety or excavate a site beyond its catchment area, or even sometimes beyond its physical boundary. Also, there are many sorts of consumption which does not leave physical evidences. So that's where our understanding is limited. And then it is very important that we use multiple proxies, as many as possible, depending on how the science is expanding everywhere and how the science is advancing. Now we can start using various different proxies to kind of supplement whatever traditional knowledge we have, right? So this is the process of interaction that we will be talking about in this particular presentation. Okay. So now, before we jump into showing you some data and making some arguments, for those who are not well aware of what was going on in Gujarat during the 3rd millennium BC. So 3rd millennium BC in Indian subcontinent is famous for its Harappan civilization, or the Indus Valley civilization. So this is kind of the shaded area is the kind of the spread of Indus Valley civilization. And then this is the specific area that we are talking about, which is the present day boundary of Gujarat. Okay, so when we talk about Indus Valley, we know there are 5 major settlements: so Harappa, Mohenjo-daro, Rakhigadi, and Dholavira. But at the same time, there are hundreds and thousands of smaller settlements which are not as big as these cities, but they also played a very crucial role in shaping the cultural expression of the Indus Valley civilization. So these settlements not only were the backbone or the pillars of this civilization, which provided this entire civilization with access to raw material, access to labor, access to other resources, say, for example, food and grains, and kind of helped shaping or helped supporting the elites, the craft producers, and the traders of this in the civilization. So when we talk about Gujarat, out of various other parts of the Indus civilization, Gujarat has been most extensively studied. Okay, so we have many sites that are being excavated, and there are sites that are undergoing excavation right now. So in Gujarat, we have 3 very different zones. So we have the Kutch, which is right here. We have the North Gujarat, which is right here. And then we have the Saurashtra region. And in this region, we also see different expression of Harappan tradition. Okay, so a regional expression of Harappan tradition. So all these red dot sites, they kind of show this very high similarities with the kind of Harappan civilization or Harappan expression that we see in the Indus alluvial region, the core region. And then all those light blue dots are indicating a tradition that is very regional to Gujarat. Now, many argue that these regional traditions that we see are the regional expression of the Harappan culture. There are some, including me, who think that these settlements, although were occupied and participate in the pan-Harappan interaction network, but I don't know to what extent it would be a good idea to call these as Harappan settlements. And although that is a separate topic of discussion, which I would not get into in this presentation, but as a gist, it is very clear that there are 2 very different traditions occupying the landscape almost around the same time. So just to give you a brief understanding of these 2 traditions before we jump into data. So we have classical Harappan type settlements where we have all the Harappan blueprints that you can think of existing. So say, for example, Harappan type bricks, both sun-dried bricks, burnt bricks, the Cardinalian direction of the settlements, very, very Harappan type architecture patterns. Although a very interesting thing that we have in Gujarat, which is not everywhere, is a huge peripheral wall around the settlements, right? Sometimes they have multiple walls separating the settlements, separating the the places within the settlement, and there are also sometimes one single wall that works as a peripheral wall of the settlement. And then at the same time, we have this Sorat tradition settlements, which are not as nicely laid out as the classical Harappan settlement. So this is a settlement of Kotarabadli, where I did most of my work. It does have a peripheral wall with very huge bastions, but if you look into the structures, it's mostly made out of stones, undressed stones, unlike many of the other classical Harappan settlements. In terms of pottery, the typical Harappan type pottery, classical Harappan type pottery that we see in the Indus core region, we also see in this classical Harappan type potteries. But when we look into the Sorat type potteries, particularly from this kind of settlements, we see a lot of these floral and faunal designs are missing, and most of them are generally geometric and banded designs. Seals and ceilings are also something that we don't have in many of these Sorat type settlements. The standardized weight system is also missing, although we have these two objects that looks like weight. It could be, it could not be, because because they were not in a sequential order, it was very hard to conclude if they were weights. Then other Harappan crafts, say for example, terracotta, so terracotta toy cars, animal figurines, gamesmen, bangles, and all sorts of terracotta cakes, different types of terracotta cakes are also missing at many of these Sorat Harappan settlements. So if you can see there, these are some of the few terracotta objects that we have found at the site of Kotarabadli. Now, there's also other very rich material inventory of classical Harappan settlements. So any Harappan settlement that you visit, which are classical in nature, you will see long charred blades, aged carnelian beads, beads of other various semi-precious stones. Sometimes lapis lazuli faience is a very common material. Shells are very common material. So this sort of typical Harappan material that you see at many classical Harappan sites are not that common in the Sorat Harappan sites. Now, it's not that it is entirely missing. So we have some classical Harappan beads. We also have some faience material, the terracotta beads, and carnelian beads. There are a lot of shell bangles and other shell products. Now, Gujarat is very famous for its shell production. So a lot of the sites have clear evidence of craft activities in terms of shell goods production as well as carnelian. At the site of Kotarabadli, we also have some copper and some very little amount of gold. So only the tip of this particular object has a layer of gold, but otherwise it's mostly copper. And just to give you a sense, this is just a fraction of Harappan materials that we find at many classical Harappan settlements. Compared to this is all the material that we have found at Kotarabadli, right? So the material representation or repertoire of these small solar settlements are not as enriched as many of these classical Harappan settlements. Okay, so when we talk about Kotarabadli, so this is where the settlement of Kotarabadli is located. So it's located at the confluence of two seasonally run rivers. This is the general layout of the settlement. So it has a central residential structure. Most of the places around the settlement is empty. And then it has a settlement wall that goes at the periphery of the settlement. Now, some argue this to be a fortification wall, as we see at many other sites. It could be a fortification wall, but it could also be not fortification wall, just because the kind of identity comes with the nomenclature. So I'm retaining myself from calling it a fortification wall, at least for this site. I would rather call it a settlement wall. Okay, and then this is again the kind of craft material that we find. Now, very interestingly, all these goods that we find at Kotarabadli are complete. So we don't have any evidence of any sort of craft production at this settlement. So craft production in the sense of that we don't see debitages that would indicate, or any kind of kiln and furnaces that would indicate that these sorts of materials were produced at this site. Rather, it's very likely that the residents from Kotarabadli acquired this material from somewhere else, and then they use those materials. And then they, at the end of their life cycle, they end up in the dump where from we found most of these objects. Okay. So now let's say in terms of what sort of data we have for us to understand human-animal interaction. And this part of the Indus settlement, or in this part of the Indus civilization. So traditionally, we have a lot of archaeozoological data, and the archaeozoological analysis of Indus civilization in general, and especially Gujarat, is very rich. Okay, there are many who have studied the zooarchaeological material from these sites and very in-depth analysis that they have done, including Professor Sharda, who is right here with us. So a very detailed data that we have. However, because of the limitation of any method that we generally use, there are only certain interpretations that we can make just by looking into the skeletal remains. So one thing that is fairly well established by now is that most of the Harappan sites has a higher concentration of cattle, buffalo compared to sheep and goat. There are primarily few types of domesticated animals, so cattle, buffalo, sheep, goat, and pigs are being the most common. So for this presentation, I'll primarily focus on cattle, buffalo, sheep, and goat, because this is what I have done. I have not looked into other domesticated animals or even the wild animals. So based on the mortality profile, so we know that these animals were consumed. We also know a lot of the cattle, buffalo, based on the marks that we found in their bones, that they were used for traction and other secondary usage. One thing that is fairly common, and then one thing that many scholars have done, is to estimate the age at death. Right. So when the animals dies would also indicate the kind of usage or kind of utility that particular animal had in the society itself. Now, not only the age at death, but also the sex. So the sex ratio of animals at certain age of death. So based on the age of death at Kotara Badli, what we have found is that 60% of the cattle, buffalo lived past their adulthood, and 30% died when they were sub-adults, 10% died when they were very young. Alternatively, looking at the sheep and goat, now I was not able to clearly distinguish cattle, buffalo, and sheep and goat based on the tooth that I had. So I have clustered them together. So based on the sheep and goat, then we see 50% of the sheep goat died fairly young, 33% when they were yearlings, and only 17% when they reached to the adulthood. So this age at death pattern clearly indicates that cattle, buffalo, and sheep goat had a very different kind of usage. Now this is a data from Bagasra. So what we see, Bagasra, Shikarpur, and Jaidak, not done by me, but one of my colleagues, Professor Brad Chase. So what they have found, this is the age at death pattern for these two sites for cattle, which clearly indicates that cattle were not used for primary products. So primary product would be hide and meat, but they were used for secondary product. Now, this data also does indicate that it is very unlikely that they were used for dairy. Okay, why? Because we don't see a lot of young male dying at a very early age, because you would not need a lot of young male if your entire economy or entire animal-based economy is based on dairy. So we clearly see that they were used for secondary products, and we have evidence looking into the animal remains that these animals were extensively used for traction, right? And then when we look at the sheep-goat data, as you can see, the sheep-goat data clearly matches with how a meat-based economy would be. So it is very likely that goats and to some extent sheep were extensively used for meat. Okay, but then this data does not necessarily indicate us that how these animals were raised, okay, and where this animal ends up, and if there are any other sorts of uses that these animals have, which does not necessarily leave clear indication or clear markers on their skeleton remains, right? So then when we looked into the isotope values of these animals from the site of Kotara Badli, what we found is that all the cattle and buffalo that were consumed at Kotara Badli had a very strong C4 signature, okay? And this is the only sheep, clearly identified sheep that we have, also lived past its adulthood, also had a very similar signature of a C4 enriched diet. Now that is not a pure C4 enriched, but it's most likely primarily C4 with some mixture of C3. Whereas sheep and goat from this area, or primarily goat in, most likely the goats in this case, had a mixed diet with mostly C3 and some occasional incorporation of C4, most likely when the grasses were fairly green. Now, this differences is very expected because cattle and buffalo are primarily grazers, so they graze on grasses, versus goats are primarily browsers, so they eat a lot of leaves. Now, because the leaves, primarily all the tree leaves, comes from C3 family, so we can see higher C3 signature, versus because a lot of the grasses, both cultivated and wild, are C4 in nature, so we can expect a C4 signature for cattle, buffalo, and sheep. Now, these are their oxygen values. So what we can see, and these are individual tooth, and this is one individual, and these are the sequential data. So it clearly indicates that a lot of these animals experience quite a drastic seasonal variation, because oxygen isotope is clearly related to the amount of rainfall on the timing of rainfall that you are getting. So we can see a very, a wet climate, and then we also see very dry climate during the time these animals were raised at the site of Kotadabadli. Now, once we look at the strontia or carbon isotope data from the site of Kotadabadli, which is right here, and when we compare with the other published data and the ongoing work that I'm doing at the site of Rakhigari, we can see the site of Rakhigari has a very strong signature of C4. Similarly to what Lightfoot also observed working in mostly in the Haryana and that part of the eastern part of the Indus civilization. These are the data from Chase et al. From Bagasara, Shikarpur, and Jeddah. And as you can see, they are very high in C4 consumption, whereas the animals from Kotadapatli is not very high. Rather, they have a strong C4 signature, but they also have some strong C3 signature. Now, based on this data, multiple arguments were made by multiple scholars. So say, for example, Brad Chase argues, primarily because he works in Gujarat, and Gujarat were predominantly indicating that these animals were predominantly indicating that Harappans were cultivating a lot of millets. So Brad Chase has argued that all the animals from this area were primarily raised on agricultural fodder in a close proximity to the settlement. Okay, so the primary idea is that they would, because they were producing a lot of millets, so the plant leftover of the millet will be stored, and then throughout the year, these animals would continue consuming these millets that are there. Now, because also just because he hasn't seen a lot of seasonal variation in terms of that, so that was pretty stagnant. Now, and similar to that was also observed by Lightfoot, although Lightfoot argues slightly differently from Chase. She argues that there are two very distinct way of raising animals. One is very typical for sheep and goat, who were generally taken out of the sediment into the wild, out of the sediment settlement into the wild, and they were eating whatever was wildly available, and that's why they have a lot of C3 signature. Whereas the cattle and buffalo were primarily raised at the settlement, having access to a lot of agricultural fodder, and then they were in a very close proximity, which is also we see for the samples from Rakhigari. Now, at Kotadapatli, I have made a slightly different argument, is that because we see a mixed C3 and C4 diet, and just because the site of Kotadapatli is located also very close to the Banini grassland, and at that time Banini grassland would be much bigger than what it is today, it was very easy for the residents of Kotadapatli to exploit the extent of grasslands that's there around the settlement, and therefore it is very likely that along with some agricultural fodder, so I did not completely disagree that there were no agricultural fodder, but at the same time there were definitely some wild fodder that these animals were consuming. Okay. So these animals were consuming. So it's a very utilitarian form of diet, depending on what sort of grasses are available at the vicinity. And I have also seen quite a bit of seasonally dependency on diet. So during the summer, when there is a lot of rainfall and a lot of grasses, then the animals were eating some C3 type of fodder, which are naturally available. And then during the winter, after the harvesting of the millets, then these animals were most likely eating a lot of millet. Now, what is very interesting, particularly for the samples from Haryana and in this region, because Haryana were predominantly cultivating wheat and barley, although they were cultivating some amount of millet, but they were also cultivating a lot of wheat and barley. But none of these animals from Haryana, including the samples from Rakhigari and the other sites, does not clearly indicate that they were also consuming a lot of agricultural fodder from wheat and barley. And it's very interesting because if these animals were fed with wheat and agricultural fodder, then why not wheat and barley? Why these residents were predominantly cultivating millets and only feeding millets? Now, there are some ethnographic evidences that sometimes millets were exclusively cultivated for the animals, but then that's fine. But when they have some plant remains, why they were not feeding their animals with those? Now, in terms of strontium data, which is very interesting, is as you can see, these are the strontium data published by Chase et al. In 2020 from Bagasra, Shikarpur, and Jaidak. So what strontium actually does is that it tells you the movement of the animal during the first 2 years of its life when the tooth was forming. So what we see at all these sites, although predominantly majority of the animals were raised locally, there are some animals that were raised somewhere else and during the early of its life, and then they were brought to these larger settlements, either as live animals or as dead animals in the form of meat, right? But this is very interesting because when we look into the strontium data from Kotadabadli, so all these solid dots and squares are the strontium data from Kotadabadli, and all these line drawings are from Bagasra. So you can see that all the samples from Kotadabadli falls in a very close proximity, except this one particular sample. But then you see a very strong scatter primarily for the cattle from Bagasra. Now, it is very likely that Bagasra, being a larger settlements predominantly occupied by craft specialists, they were most likely acquiring meat from somewhere else or the animals from somewhere else. But it's also possible, just because Bagasra being a larger settlement, there were higher movement of human, possibly with animals. So it's good possible that some women were getting married to the site of Bagasra, were bringing some animals from their parental home, or vice versa, if the males were getting married to Bagasra and bringing animals. So being gender neutral in this case, were most likely were also bringing some animals with them. It is very possible, but it is also possible, they were acquiring animals and other food resources from multiple other settlements. But for Kotadabadli, being it a very small settlement, is very likely that all the animals, that it could be possible that they were self-sufficient, and all the animals that they consumed were raised by them, and therefore were raised in a very close proximity to the settlement. Now, so we have clearly seen so far that cattle, buffalo were raised differently compared to sheep and goat. And we have also seen that not all the settlements raise their animal in a very same or very distinctive way. There are variations. There are also regional variations, right? Now the question comes, well, we know cattle, buffalo, and sheep, goat were consumed for meat. We also know that cattle, buffalo were consumed for secondary products, primarily traction. The one question that remains to be answered, if these animals were consumed for dairy, because dairy is a very important industry when we think of archaeological settlements, because it helps to generate surplus. Okay, now how is that? If you primarily depend on meat from animals, you have to kill the animal to acquire the meat. So if you want to increase your surplus based on meat, you need a very large herd, and it also gets very difficult to manage the herd or to maintain the exact amount of herd, specifically in the bad climates, when they're dry, and then there are disease and things like that. So there's a very high chance that some climatic condition or some natural condition, natural discrepancies in your herd will deplete exponentially. But on the other hand, if you have subsistence, if you have an economy that depends on dairy, now you can increase surplus, primarily because you don't have to kill your animal and you can exploit dairy from an adult cow for many years, but also that you can convert your dairy into other dairy related products, which would also increase the shelf life of this particular product. So say, for example, a lot of you have ghee in your home, and you can see that, you can keep a ghee for a few years without it getting spoiled. Butter can be stored for a very long time. Even some cheese can be stored for a very long time, soft cheese and hard cheese. So as you can see that, you can increase the shelf life, and you can also increase the amount of surplus that you have, and you can easily use this surplus for any sorts of trade and exchanges. So that's what I wanted to see if there are a clear sign of dairy at this settlement. So what I have found is that looking into the residues that are there in the archaeological vessels, and okay, so just to give you a brief idea of what these food residues are. So most of the pottery vessels that were used during the Harappan time were perforated, has small pores, not perforated, has small pores, and they were not slipped or they were not glazed. Okay, so what is a glaze? Like if you see porcelain utensils at your home right now, they're heavily glazed, so they don't have those small pores to absorb the food. However, on the other hand, the Harappan ceramics had a lot of pores. So anything liquids in a liquid form that gets in contact with these vessels, the pores of the vessels will absorb those liquids. Okay, and sometimes the pores are so small that even it is hard for the bacteria to get into those pores and degrade whatever organic materials are there. And these pores, also the matrix and the lipids, they create a polar bond, which sometimes prevents the lipids to get washed out of the pottery because of the water. At the same time, as we all know, lipids are hydrophobic. So have you tried mixing water with oil? It does not, because oil are hydrophobic, so they don't get dissolved in water. Some low chain lipids are not hydrophobic, so they get washed out of the pottery, but majority of the lipids, they don't get washed out. And therefore, potteries are a great source to find lipid residues. Okay, so when you extract these lipids, you can possibly interpret, depending on the preservation, what sort of food that were processed in those archaeological vessels. Okay, so lipids are basically fatty acids. So you can think of oil, fat, wax, and all these things would fall under the category of lipids. Okay, now what we have found is that a lot of these vessels that were used at Kottatabadli had clear indication of meat, okay, coming from different animals. So we have clear indicator of ruminant meat as well as non-ruminant meat. So what would be the ruminant meat? Primarily sheep, goat, cattle, buffalo, who has a rumen, they would be called as ruminant meat. And then you have birds and pig that would fall under non-ruminant meat. So we have strong indication of non-ruminant meat here, and we have strong indication of ruminant meat right here. And the other interesting find is that a lot of these vessels that were there at Kottatabadli indicated dominance of dairy. Okay, so why I say dairy and not clearly say milk? Because it's very hard to distinguish what form of dairy it is, whether it's a milk, whether it's a ghee, whether it's a cheese, whether it's a butter, but any dairy product for that matter. And a lot of this dairy, and so this is the general what we see: a lot of meat and a lot of dairy. Now you can see that a lot of this meat, majority of it, were coming from animals that had a pure C3 diet or a C3 rich diet. And if you remember the isotope data that I have shown you, is that those are the goats and sheep, primarily the goats. So a lot of the meat were coming from goat, and which is also expected because of the archaeological data that we have seen and how the goat were slaughtered at this particular settlement. And then we see another group of animals which had a predominantly C4 type of diet. Now, what we have seen, the cattle and buffalo had a primarily C4 type of diet. So we see some meat that were definitely coming from cattle, buffalo, but the entire dairy that we see at Kottatabadli were exclusively coming from cattle and buffalo, and nothing was coming from sheep and goat. Now, the question remains why? Because sheep and goat can also produce dairy. Then why were these people at the settlement of Kottatabadli were only exploiting dairy from cattle and buffalo, but not from sheep and goat? Now, one of my colleagues also analyzed this data, analyzed data from other Harappan settlements, both in Gujarat and Haryana, and she has published it in 2021. And she has also found that all her meat, primarily all the meat, were coming from animals that were enriched in C3 type of diet. Now, we don't have, now, although a lot of her data plots near the non-ruminant category, but we know that, the non-ruminant were not that much. So it's just because of some mixing and some regional variation of isotope values that there's a shift in this data. But if you look into the C4 area, that you don't see a lot of cattle meat right here, but all the dairy that you see are coming from C4 type animals, so cattle and buffalo. Now these are the two modern dairy data, just to show that the method actually works. So it's a similar finding that we both found that the entire dairy, the entire batch of dairy at different sites, not only at Kota Dabali, but even at different sites, were primarily coming from cattle and buffalo. Now, when we look into the dairy data, what we found that dairy were boiled. So we found dairy residues in cooking vessels. We also found dairy residues in a lot of bowls. So primarily, majority of the bowls that we have analyzed had a strong dairy residue. So there are 8 bowls, and as you can see, 4 of them has dairy and 4 of them has meat. Now, these bowls indicate that some of them dairy was probably consumed as milk. So you can pour the milk in the bowl and drink it. They may have made porridges out of milk. They could have also made other milk products which are liquid to semi-liquid in the nature. So they used bowl to consume those dairy. Then you also have a pot that was also used for the consumption of dairy or the storage of dairy. Then we also have ladles. So these are a bowl-like ladle. So it has a bowl at the end, and then it has a ladle, like kind of a thing. So some people, they call it a ladle. Some people, they call it a handled semi-spherical bowl. So whichever way you call it. So these bowls also had some indication of dairy residue. And interestingly, the perforated jars, although I have only analyzed one perforated jar because there were not many at the site of Kota Dabali. So I've analyzed one perforated jar, which also indicates the presence of dairy. So it's very likely that these perforated jars were used to most likely to strain water out of cheese. So once you break the milk and then you get the solid milk solid, and then you have to drain out the whey. So that's most likely what they were using it for. And then we have meat. So we have bowls and a big basin type jar right here, which indicates the consumption of meat. So these are most likely tables where some form of meat stew were served, both using beef as well as mutton. Okay, so maybe the very early history of mutton gravy that we have today, maybe in a rudimentary form, were also consumed by the Harappans. Now this is a big basin. So I think that this big vessel was most likely to use maybe to marinate pork or any bird meat, because this is a non-ruminant meat, or it was a serving vessel where they processed a lot of this meat and then they subdivided it into smaller vessel. Okay. And then we also have indication of ladle, which indicates that they were also used to pour some of this meat gravy, right? So this is, you can think of this like the big spoons that we have, and then you just dip the spoon and then serve it. And then there are definitely some cooking vessels. So all the squares and rectangles are the cooking vessels. So definitely the meat were cooked in the form of a stew, and that's why they used a lot of cooking vessels. Now, the question is, you might think that, why we have like so many cattle bones scattered across the settlements, and every settlement, why there are not so much cattle or beef residue in a lot of these vessels? Well, there could be multiple reasons for that. One would be that the sample size is not big enough. So I have analyzed around 59 samples, and Akshata and also analyzed a few hundred samples. So the sample size is not big. So we may have missed a lot of beef residues just because of the sample bias. But it could also be possible that mutton were most likely and some beef were most likely processed in the form of a stew or a gravy, where a lot of beef were processed, most likely in the form of other ways, say, for example, roasting. Now, Brad Chase has a very nice argument for that, and I also think that it could be a valid argument, is that if you slaughtered a goat, particularly if the goat was fairly young, so it did not reach the complete adult size, it produces some less amount of meat compared to when you slaughter a cow or a buffalo. So in order to consume a cow or buffalo in the absence of a refrigerator, you need to have more people so that you don't waste the food. Similarly, one family or one extended family or a couple of family could easily consume one goat if it was slaughtered. So it is very likely that cattle were most likely, or cattle buffalo were most likely slaughtered only in a very specific, most likely ritualistic or ceremonial context, where there were more people who could consume the cattle. Okay, and that's one of the reasons where we don't see them in a day-to-day utilitarian vessels, right? Now, so now we know how these primary two types of domesticated animals were raised and how these two types of domesticated animals were consumed. Now the question comes, how were they discarded? Okay, now at Harappan site, in most of the Harappan sites, we don't clearly see that, cattle were discarded in a different way versus goats were discarded in a different way. Maybe if Sharda knows, she would be able to add on to that. But there is no a very distinctive way. But there is a very interesting finding. Okay, and the interesting finding was that when we looked at the site of Kotarabadli, we found a huge dump of ash around the wall of the settlement, right? So as you can see, all these layers right here, they are fine ash with all the other garbage that includes cattle bone, sheep goat bone, pottery, all the artifacts and everything that you can think of. So they were dumping a lot of these domestic waste or any sorts of waste along this settlement wall. But then where this ash is coming from, because we don't see necessarily craft production and craft activity at the site of Kotarabadli. So then what was the source of this ash, and why were there so much of ash if you think that this was the practice across the entire settlement wall, which is close to a hectare big? So that's a lot of ash, right? And the current surviving height is, I think, 1 meter something. So that's a lot of ash, right? And then when you look into the settlement area, so that's the occupational layer right here, and you don't see this ash. It's very neat and clean. So all the ash is along the settlement wall. Now, not only at Kotarabadli, but there are also other sites where you see around the time when the Sorat occupation takes the dominance, we see this kind of ash. Okay. And when we analyze the ash, so we saw a lot of phytoliths in the ash. We don't find a lot of big charcoal, so that means there were not a lot of wood that were burned. And looking into the present environment at Kutch, it is very unlikely that they had huge trees, which would produce a lot of wood for them to use as fuel. So they were most likely using grasses and other material that are there to as a fuel, and therefore we see a lot of grass, and the majority of them were coming from Panicoidea and Poety. One interesting find is that not only they had a lot of grass phytoliths, but all this ash that were there around the settlement wall also has a lot of spherulites. So what are the spherulites? Spherulites are the silica bodies that are formed in the guts of the animals, and then they come out with the feces that the animal produce. Now, if you dump feces or burn feces and dump the ash, you would also incorporate a lot of these spherulites into the sediment. And this is exactly what we're seeing, that, not only that all these samples from the ash have a lot of grass, but they also have a lot of spherulites. Now, if you remember that the cattle from the settlement was consuming the grasses that are available around the vicinity of settlement, so we expect a lot of grass if they were burning the dung produced by these cattle. And because they were burning dung, we also expect a lot of spherulite. The other thing that we also see is that all the samples from Kotarabadli has, all the ash samples has a higher concentration of nitrogen. So the primary source of nitrogen in the sediment are from organic matters. Okay, and now because it is ash, so there are no, and the And just the decomposition of plant bodies does not leave a lot of nitrogen, but if you have a lot of dung, it leaves a lot of nitrogen in the sediment. And this is exactly what we see, that all the sediment from the ash has a lot of nitrogen compared to the residential area as well as the pre-occupational layer from Kotadabadli. So it's very unique that, they are burning this dung either as domestic fuel, like the dung cake that we currently burn, but it's also that, because they're raising all these animals and they're producing a lot of dung, and then they were disposing of this dung around the settlement wall, and then they were burning periodically to maintain the hygiene of this settlement. Now, this is a very unique character of this particular settlement and some other sort of settlements from Gujarat, but we don't see that clear sign at some other settlements. So we also see that animal dungs were treated slightly differently, most likely coming out of from cattle and buffalo. They are also treated something differently. So it's not only that cattle were raised differently, they were consumed differently, but some other remains of these animals were also treated slightly differently. Okay, so now at the very end, so what have we observed? So we know that cattle and buffalo played an important role in the Harappan civilization, because not only they were the majority of the animals found at any Harappan sites, but we saw depiction of these animals in various forms of, in various ways. So in the potteries, we saw them depicted in the seals. So cattle and buffalo played a very major role. Human interaction with cattle and buffalo was also different than the nature of interaction with other domesticated animals, such as sheep, goat, and pig. And then, as we have discussed by looking into the isotope values, that, and also that, the cattle were the primary animals that we got from different areas. So different cattle from different areas were moving. But at the same time, the sheep and goat and pig were primarily locally domesticated, and they were availably local, right? They were there at the settlements, and not a lot of movement of sheep and goat and pig that we observed. The nature of interaction slightly varied between regions and most likely between regional cultures. Now, this is very interesting, and this is something that is likely, and we don't have a clear answer to that, is that these animals and the way these animals were raised and consumed at some other Harappan site, it was not clearly the same at the settlement of Kotadabadli. Some sites we see a very strong dominance of C4 fodder for cattle foddering, irrespective of their primary cultivation type, right? Even in the Haryana, we see dominance of C4. Even in Gujarat, we see dominance of C4. Whereas at the site of Kotadabadli, we not only see C4, but we also see some regionally available C3 grasses that, or some C3 bushes and plants that these animals were consuming. So it's also, there is slightly difference the way, these settlements, these larger craft producing settlements were raising their animals, the way that they're raising their animals versus the way these agro-pastoral settlements were raising their animals. Then specialized production of animal as part of Harappan economy that supported the full-time craft specialization during the urban period. Now, this is very interesting because by the time we see the urban Harappan period, we don't see, the hobbyist craft producers. So we see very standardized, well-developed, advanced craft production, most likely people engaging in these activities throughout the year. Okay, so they are full-time craft specialists. Now, once you have this sort of full-time craft specialists living in a settlement, I think it is very important for them to have access to food, because you don't expect someone making a bead and then in the morning, and then, or herding their animals in the morning and making beads in the specialized East Carnelian bead in the evening. It's most likely that they were specialized in those activities, and it's very likely that there were other settlements, nearby rural settlements, who were providing them with not only animal product, but also plant product. And that's also very interesting because, once we start, capitalizing on dairy, which you can extract frequently as well as store for a longer period of time, this sort of interaction becomes fairly easy, right? So this sort of interaction, a whole scale interaction that we're seeing for the Harappan civilization in terms of how humans were interacting with cattle, buffalo, and sheep goat in both day-to-day utilitarian use, a commercial economic use, as well as symbolic use. Now, obviously, because there are only a handful of these sorts of analysis available for the Harappan civilization, whether it's India or Pakistan or any other part, our understanding is very limited, and it's primarily concentrated on these few sites altogether with a very limited number of samples. So in order to develop a better understanding of how this interaction developed to the extent that we see during the Harappan civilization, how that continued and shaped the way that we see in the present culture versus how it was in the very beginning era needs to be analyzed for us to understand the shifting dynamics of this interaction and how changing economic condition, changing environmental condition, changing political condition might have shaped this sort of interaction. And even today, it's very interesting that we see that, cattle plays a very unique role, right? So cattle is a both symbolic, ideological animal, as well as is also very utilitarian animals. But at the same time, there are only certain things you would exploit from cattle, considering the present society, and certain things you would not, right? So you would exploit cattle for their strength as a traction animal. You'd also exploit dairy from the cattle, but you would not exploit meat from the cattle for whatever reason, social, political, religious, ritualistic, whatever reason. So this sort of understanding and the shifting dynamics of how humans were interacting with animals and animal products needs to be developed for us to understand how these complex societies were very early from this present time, around 3rd, 4th, 5th millennium BC, performed and interacted and survived and developed into one of the largest civilization of world, one of the most sophisticated urban civilization of the world is very important. So thank you for listening. I don't know if I have crossed the time, but most likely not. So yeah, open to question answer.
Well, thank you for this wonderful talk, Dr. Kalyan. It was indeed well within time, and it's always fascinating to listen into various aspects. Like, it is not only the material culture, but all those intangible aspects which can come through with this kind of scientific analysis. Now I will request Sharda to take over and moderate the Q&A session.
Thank you. So I request all the participants to type their questions, comments in the Q&A box, not in the chat box. And so I see one question right now. Okay, I'll ask that, and then I have 1 or 2 questions. And I think by then it'll give us the other participants time to type in their questions as well. So this is from SK Aruni. And it's slightly different. The question doesn't concern animals as such, but some other aspect of the Harappan civilization. So he asks, the large settlements have enclosed walls. I think this is in context to you were talking about classical versus Sorath.
Yeah.
The large settlements have enclosed walls. What were the functions of these enclosures? And should we equate with fortified towns of the historical period? But the enclosed walls were not necessarily built with raised bastions and watchtowers, is his question.
Okay, so it's very interesting that it's not only the large settlements in Gujarat, but small settlements like Kotara Badli. As you can see, it's a very small settlement, also has these wall enclosures. Now, should we think that it is similar to the fortified towns of historic and medieval period? I would say that just because there are some structural similarities, if we think that that they were similar to historic period, it will be slightly far-fetched. And I'll give you some indication. So if you look in the Egyptian part of the world and the Mesopotamian part of the world, we see enclosed settlements fairly early. We see Neolithic enclosed settlements. We also see settlements from very early Chalcolithic, before the empire period that sets in Egypt and Mesopotamia. So we see enclosures. Now, The bastions, as they have been identified as bastions. So whether they were bastions or whether they were support pillars for these walls to survive in an area, whatever for whatever reason, is hard to say. Now, I am not completely denying the fact that they were not fortification and they were not built to prevent some sort of external threat, but we don't have a clear sign of any sort of external threat that these walls were protecting the residents from. It could be that these walls were protecting whatever things were there inside, rather than having a very exposed settlement, which is open from all the sides. So it gives some sense of security. Having a wall around you gives you some sense of security. It's not necessarily it's fortifying the settlement. It gives you some sense of security. It keeps things within the settlement. You can also maintain who gets in and gets out of the settlement. It also provides some sort of a security from wind. It provides some sort of security from flood. So all these sorts of functions are also there. So it's not necessarily a fortification of a historical sense, but then, yeah, definitely there is not enough data to either prove it or disprove it. So that's why I prefer to keep it open rather than giving a very strict nomenclature to it that this is fortification. Because when I say fortification, it comes with a lot of burden associated with that term, that, okay, this fortified settlement, there's something, very unique happening inside the settlement. There were threats from outside. There were people, there were watchtower, there were people moving around the settlement while saving whatever is there is inside. So that sort of military, military interpretation of a structure is slightly problematic, and I think we need more data for that.
Okay, so the next question is from Ahana. She asks, what are some of the major criteria that you have considered before choosing the different ceramic functional groups for Kothada Bagdi?
Okay. So ceramic functional groups, my idea was to represent all the kinds of ceramics that are there at the site of Kothada Bagdi, rather than picking and choosing. No, there's no harm in picking and choosing if you have some vessels in very specific context. So let's say you have a storage jar that's in a very specific context. You have some pots intact in a very specific context. So if you have those sorts of samples, then yeah, it's definitely a good idea to pick and choose, but we didn't have those sorts of things at Kothada Bagdi. So my basic idea was, rather than focusing on cooking vessels or storage vessels, let's have a wide sense of what things were happening. And that's why I have analyzed close to 60 ceramics, and only 22 ceramics produced enough residues that I could do isotope analysis on. So a lot of these samples had no residues or very low concentration of residues. Sometimes I have experience with working with cooking vessels. They are sometimes very good for residue analysis, sometimes very bad for residue analysis, depending on what part of the vessels that you are getting. So I think it is more important to think of the part of the vessel you are analyzing rather than the kind of vessel you are analyzing. If you don't know anything about the function of the vessels, go for everything.
Okay. So the next question is by Roshan Paladugu. I hope I'm pronouncing your name correctly. And the question is, does the presence of animal dung ash in sediments increase the stable nitrogen isotope ratio along with the absolute concentration of nitrogenous compounds? Okay.
So stable carbon nitrogen ratio, so the nitrogen isotope ratio increases with the incorporation of dung, because that's a great source of nitrogen, not only in the sediment, but even sometimes if a field is foddered with animal dung, we see increased nitrogen isotope values even in the grains as well, even in the plants as well that they're growing. Now, the absolute concentration of nitrogen in the sediment, so I have not measured, did I measure? The absolute concentration of nitrogen values would also increase. So not only the isotope values would increase, but the absolute concentration of nitrogen would also increase. So the bulk nitrogen, if you analyze, the bulk carbon that you analyze, that would also increase with the sediments being incorporated with dung, because all the sediments, I did not show you the figure, but all the sediments coming from the residential area and the unoccupied layer indicate very low concentration of nitrogen, as well as slightly enriched C4 nitrogen, C4 carbon, compared to all the ashy sediments that have nitrogen in there. So they're more slightly shifted towards C3 because we have grasses from variety of regions, as well as dung from maybe from sheep, goat as well, and other animals as well. So it shifted slightly towards C3, and then it also shifted slightly enriched with the nitrogen just because it has a lot of dung.
Okay. So the next question is by Ravikant Prasad, who asks, who thanks you for the talk, and then asks how accurate can we, like through isotopes, interpret what sort of or what different purposes a particular animal has been used for. So I will try to add my question to this because it's kind of bordering on the same thing, that unlike the hard animal parts, which survive very well, the bones, teeth, shells, horns, etc., we are talking of biomolecules here, which are very, very, sensitive to degradation. They can degrade easily in varying temperatures, pH of the soil or water changes. They may also harm, not harm, as in they may destroy these biomolecules or change their composition, right? So first of all, why do we assume that these biomolecules are so their integrity is so well preserved through time and through all these biostratinomic and diagenetic changes? And second thing is, how do we factor in these, if at all there are modifications in isotopic signatures or say proteins or DNA, how do we take care of these changes that happen over time through conceptually and through the methodology that we follow for extracting them, first for analyzing them, and finally for interpreting them?
Okay. So first let me answer the biomolecular part of it before jumping into the isotopic part of it. So I have tried extracting proteins from ceramic vessels. I have failed miserably just because the protein did not survive in any of that. So what I ended up finding a lot of human contaminant proteins, so a lot of hemoglobin, a lot of sweat proteins that are generally there. Now, that could be archaeological, but that could also be modern, and it's very hard to distinguish in that case when you have similar sort of protein signature both inside the ceramic versus outside the ceramic. It's most likely coming from the contamination. In general, across the world, there are very few studies or very few instances where they were successfully able to extract proteins from ceramic vessels. Okay, so amino acids from ceramic vessels. No, I'm not talking about amino acids from any other sources, not collagen, from ceramic vessels. It's very hard. Now, what we have seen in general, that lipids generally preserves better compared to proteins, and carbohydrates obviously doesn't preserve. So the only ideal source would be to look into starch because the carbohydrate as a chemical would not preserve. So lipids, just because they are hydrophobic in nature, so one thing that it generally preserves, but one major issue with lipid is that there are bacterial lipids. Now, bacterial lipids are very similar to what you would expect for archaeological lipids. So they are very similar. It's very hard to distinguish that whether it's a bacterial lipid or it's coming from the food residues. Okay, now that's a major problem. The other issue is the leaching of other lipids from the sediments and getting into the pottery. Okay, just because, and that we have seen not only for fatty acids, but we have also seen for alkanes that they're prone to leaching. They don't leach so much just because they don't get dissolved in water. So they don't leach so much. But then there could be exogenous lipids on the immediate surface of the pottery. Now, how we distinguish and how we see if there are exogenous lipids. So say, for example, there are a few things is that we also analyze as a control the outer surface of a ceramic vessel, not those that were used for cooking, because if you use those that were used for cooking, you will find a lot of hydrocarbons, which are most likely coming from the fuel that were used. Okay, so leave those, but if you take other ceramics and look into the outside lipid and see the inside lipid, and if you see there is a clear difference that the concentration is much higher versus the low concentration. Now, some lipids could percolate all the way through the outside from the inside during the cooking. And in that case, the distribution would be slightly different. The concentration would be slightly different. Also, we generally don't take any lipid below 20 microgram per gram of sample and interpret them. Okay, so I would generally just say that, these lipids indicates high dependence of animal origin, mixed animal plant origin, or fish origin if there are biomarkers and things like that. I would never take them for isotopic analysis. Okay, some people argue that they generally prefer samples more than 5 microgram per gram of lipids. Okay, if they have that, then you can take it for isotopic analysis, but I tend to take only those samples that produce high concentration of lipid. So the other control is if you have multiple charts from one location, and if they all produce same lipid data of different vessels producing same lipid data, then you know that they are coming from contamination. But if you have samples close by to each other, one had a lot of lipid, one doesn't have any lipid, so then you know that it's not contamination. Also, for isotopic data from the biomolecules, we tend not to use lipids that are easy to get incorporated. Okay, so what are those? So those are some large lipids. So say, for example, sterols. So we would not do isotope analysis of cholesterol or sigmasterol or phytosterol. Even if we have them in archaeological data, if they are not degraded, if there are no cholesterol byproduct but the true cholesterol, we would not interpret that, we found cholesterol, so that means there are animal products, because cholesterol is something that can easily get incorporated as contamination. So as phytosterol. So we would not do that. We would not analyze the isotope values of low chain lipids because they can easily get dissolved in water, which would not be there in the archaeological. So that can get contaminated. So we wouldn't do that. We wouldn't also do that and then there are certain lipids that would clearly indicate there are contaminations, right? So say, for example, if there are, as I said, non-degraded cholesterol and phytosterols contamination. If there are very high concentration of unsaturated fatty acid contamination, because such unsaturated fatty acid which at some point would degrade. Okay, if there are some fatty acids which are very unlikely to preserve. So let's say you have a very low concentration of low and mid chain fatty acids and very high concentration of long chain fatty acids. That's most likely coming from contamination. There are other contaminations. So there are plasticizers as contamination. There are some biomarker contaminations, say, for example, homostanol, which is a contamination from sunscreen. There are DDT, DDH, and all sorts of plasticizer. There are contamination from ink, so octanomic sulfur. So if you see a lot of octanomic sulfur, they're coming from contamination. So if you see all those contaminations coming in your samples, then you don't use those, then you discard those. But then there are also, say, for example, dicarboxylic acid, which are not present in the nature as a free dicarboxylic acid. So they could only get produced if a unsaturated fatty acid is heated at certain temperature. So if you find those on your lipid data, within your lipid data, then you know that that's coming from the food sources and not coming from the contamination, because you wouldn't find them in general environment, right? Let's say you are finding a lot of alkanes, and then you're finding a lot of low chain alkanes to mid chain alkanes, then they are most likely coming from contamination. Okay, they are not coming from the original source. But if you are finding higher chain alkanes, then they are in the plant, so they are not contamination. That's also true for, if you don't think of alkanes, alcohols are some good examples. So if you find alcohols, then in a degraded condition, then they're coming from waxes. But if you're archaeological pot and you find a lot of waxes, which is very old archaeological pot, and you don't find alcohols, and you only find like intact waxes, then that's most likely contamination. So there are multiple ways that you determine if the data that you are getting is contaminated or not contaminated. Only you are certain that it is not contaminated, you send them for isotopic analysis. And that's why out of 60 samples, I've only sent 22 samples and not all the other samples. And there are
So,
other lipid.
From Rakhi-Gadi, how many samples do you have? Because you were mentioning that you have started work on Rakhi-Gadi and you showed some results.
So that's the animal isotope. So that's the that's the enamel isotope. That's not the lipid residue. So I don't have I have not yet analyzed any samples from Rakhi-Gadi for lipid residues. So no data.
Okay.
Now in terms of isotopes, okay. So
generally we know of like oxygen, nitrogen, carbon, strontium, but besides that, any other isotope, and what would it tell us about the animals in the past?
Okay, so there are a lot of isotopes that you can work with. So you can obviously work with oxygen, carbon, nitrogen, strontium. These are fairly common. You can work with sulfur isotopes also to identify with what kind of animal or what kind of species that you're dealing with in terms of its pyramid. So the it's a vertebrate versus non-vertebrate, but what sort of thing. So that can be done. Lead isotope is another good isotope. So if you combine lead and strontium, that would give you a better idea of migration rather than just strontium alone. So that's a good isotope that you can use. So these are the primary isotopes that we generally use in archaeology. Most common is obviously carbon, oxygen, and strontium. Now, oxygen isotope, there is an argument that because the phosphate in the tooth is much stronger compared to the carbonate, so the carbonate gets contaminated, degraded, or get diagenetic alteration is much higher for carbonate, but it is not for phosphate. So the phosphate is the ideal source for oxygen isotopes. But there are studies that have clearly indicated using fossil samples, not even archaeological samples, that the diagenetic alteration is not that high, that, you know, if you use carbon oxygen isotopes from carbonate, you would get completely different result. So it is not like that. Also, if you want to compare carbon and oxygen, then you can only go the carbonate pathway, you cannot go to the phosphate pathway. And the carbon isotopes we have seen is fairly good. Even like very old fossils also have produced very reliable data. And you can always do a general FTIR scan of your tooth just to see, you know, what sort of preservation are we talking about? Are they degraded? The things get complicated when we're extracting isotopes from organic matters. So all the carbonate and phosphate, they're inorganic source of isotope. Now, when it comes to organic, that's the collagen, that gets trickier, because sometimes I have seen, and I have presented last time as well, that not all the tooth produce enough collagen. Sometimes they had like no collagen preservation, completely gone. Sometimes they have some amount of collagen preservation. So you can actually use the collagen to get bulk carbon isotope, but you can also use the collagen for to get the compound specific isotopes from amino acids. Okay, right. So then also amino acid, you can choose the specific the species specific amino acids, which then you know that they were not coming from somewhere else, but from the animal itself. So there are many ways. Like if you're thinking of contamination, we know now that there are many ways that you can.
So we have characterized them now and well enough to know that we can immediately remove them from the results.
Yeah, exactly. It is very well developed now. There's no question of the authenticity of the result.
Okay. Yeah, I think this last question is kind of more or less answered now. No, so very quickly, if you can just answer this, it's by the same participant, Ravikant, who says that the samples, plants or animals may show the carbon isotope values which are overlapping with the C3 and C4 plants. What sort of interpretation can be made from such carbon isotope values?
Okay. So overlapping C3, C4 is more like it is a mixed C3, C4 signature. Now, there are geographic variations, there are regional variations in carbon isotopes, there are seasonal variation in carbon isotopes as well. So you wouldn't expect us. So if you feed the animal with seasonally varied fodder, all C4, you would still not get a straight carbon line. It would still vary with the season. Okay. Now, when you get a mixed isotope values, then you know that this is a mixed data. Okay. Now, some people would go and identify the percentage of C3 and C4 in their diet based on the isotope data. I don't prefer doing that, particularly in Indian context, because we don't have very strong reference collection. If we know the regional variation, if we know the climatic variation of these values fairly well for every region, then you can go, that 30% of C3 versus 70% of C4, 30% of C4 versus 70%, all those sorts of characterizations. If you have very well developed reference collection, if you don't, then just call it mix. That's what I prefer to call it. I just call it mix with higher concentration of C4 over C3.
Okay, I have one last question, which if you can just quickly answer.
Yeah.
So you very correctly mentioned that sheep and goat have different diets, they're grazers versus browsers versus grazers. So through morphometrics, we can, if we have a good large data set, you were talking about around 20-25 samples. Even with that, I guess, morphometrically speaking, we can kind of distinguish between sheep versus goat. Now there are enough published keys available to distinguish based on shape and the measurements of individual molars and cusps of molars. So why did you not attempt that? Because you said you clumped them as sheep goat.
Yeah.
Because I understand they're very closely related. It's impossible to distinguish between them unless we use a caliper or look at them close enough to find out the shapes. But it is still kind of possible, and that would have brought in more nuanced differentiation and ultimately interpretation, where you could clearly say sheep had this isotopic values and goat had these isotopic values. So did you look at any of these like morphometric possibilities?
Yes. So when I collected the samples, I just collected sheep, goat, cattle, buffalo. Okay, so not sitting with the material and then specifically choosing cattle, buffalo, cattle and separating buffalo, separating sheep versus separating goats. Just because, it's not that always you plan things and it falls in a, so I had only few days to sample. And then that's what I just took whatever I got and then went back to Toronto. Now, once I was there, I tried to identify the sheep, goat, and cattle, buffalo. And I was fairly confident with that particular sample that I identified as sheep, just because on whatever published material was there. Now, I did not necessarily go and look at the reference collection available at University of Toronto just to identify, just because they would not have the similar reference, the region-specific reference collection. I don't think they have any reference collection from India in general. So I did not do that, but I was fairly confident identifying that one particular tooth is highly likely of sheep, and the rest were more likely goat. But then just because my capacity of identifying them, like I don't identify myself as an archaeozoologist, so I am not confident with my identification of that all the remaining are goat. So I clumped them together as sheep, goat, just to be on the safer side. Now, by looking into the isotope values, it's very likely that my identification was right. There was only one sheep and rest all were goat, just because they have the similar isotope value. But I prefer not to do that. If you were there or Pankaj were there or Joplekar sir was there, I would have probably showed that.
Because that's the first question that comes to our mind, that it is maybe sometimes possible to distinguish, but no,
it's possible, and with my capacity, I could only distinguish that one tooth.
But it's still fine to kind of keep them in larger groups, like sheep, goat, or cattle.
Yeah. So other ones I was not 100% sure, so I clumped them together. One I was sure, so I put it separate.
Okay. All right. So I don't see any other questions for now, and I think you've answered all of them very patiently and in good detail. It's also a learning experience for us. These are very new methods that many of us are not so familiar with, and even reading, even through reading, it's not easy to assimilate or understand. So thank you. Prabhakar?
Yeah, thank you so much for this wonderful talk, Kalyan, and really, I think this field has to be developed a lot, more and more of such studies, and through a longer period of time. You were telling about a single particular time span, right? But like an urban center like Dholavira, if it can be done, that would give wonderful insights.
More samples. As much as we can, we'll have much higher resolution of a lot of questions are just hypothetical right now. A lot of answers are hypothetical right now, but more samples, more data, more robust data, not only archaeological, not only Harappan, but starting from the very early occupation to
Yeah,
all the way
yeah.
modern, that sort of resolution we need, which is there for Europe. Like they have such a huge data. So we don't like, so. Well,
maybe slowly it will develop. Slowly,
Yeah.
bits and pieces, different regions. If we start the work, then it will slowly develop, I think. Anyways, thank you. Thank you for this wonderful talk and for the time you have given us. And I look forward. I also thank all the participants and I look forward for the participation in the forthcoming lectures also. And we close here. Thank you.
Thank you. Thank you everyone for
Thank you.
listening.